A number of structural complexes join individual epithelial cells together near the apical surface. They're collectively termed the junctional complex, and there are three types. They can really only be observed when viewing high magnification electron microscope images of adjoining cells. I'm only going to provide a general summary of these three types of junctions and also show a couple of examples where a specific junction is present in other cells besides epithelial cells. Let's revisit this inserted image taken using an electron microscope of the apical surface of these enterocytes along here. Here's the nucleus of one and the nucleus of its neighbour. Here's the brush border, and if you look very closely, you may be able to see a very thin line passing between the cells near the apex. Now look at the electron micrograph image. The microvilli you can see belong to two adjacent cells. The microvilli of one cell is along this apical surface here. The other cell surface is in this small corner region of the image here. And that thin line I mentioned a moment ago runs down between the two cells, which are the two lateral cell membrane surfaces, and so one lateral border between the two cells. Just here is the zonular occludens, or tight junction. This occluding junction, or tight junction, forms an impermeable diffusion barrier so that any molecules, including water, must pass through the cell first to enter the subepithelial tissues. As the arrow indicates, this junction is located at the most apical region between the two adjoining cells. And this junction also prevents the movement of cell membrane components between the apical and lateral cell surface domains. Just below is a rather dense area where anchoring junctions link the cytoskeleton of the two adjacent cells and so maintains structural or mechanical stability. They can also be located at the basal domain of cells. One type of anchoring junction is the zonular adherens, which interacts with the actin filaments inside the cell. Remember, along here are the actin filaments belonging to the terminal web to connect and support the actin filaments within the microvilli. Another anchoring junction is this dense spot here. It's called a macular adherens or a desmosome, and it links the cytoskeletons of the two adjacent cells. Desmosomes are very prominent between the keratinocytes of the epidermis to strengthen this epithelium so it can withstand some very considerable shear forces. Another structural version of the desmosome, called a hemidesmosome, anchors the basal areas of the epithelial cell to the underlying connective tissue. Communicating junctions, or gap junctions, permit the diffusion of small molecules between adjacent cells. This then allows intercellular communication and so coordinated cellular activity. Here's a sheet of smooth muscle in the wall of the gut. The smooth muscle cells are sectioned longitudinally here, and so you can see their elongated nuclei. But another layer of smooth muscle is sectioned transversely down here. All these smooth muscle cells are connected by gap junctions, and so ions can pass between the cells and so can regulate the contraction of this entire sheet of smooth muscle. Gap junctions are also very important in cardiac muscle cells and are located at the sites where individual cardiomyocytes are anchored together. The intercalated disc is the attachment site for adjacent cardiomyocytes, and the presence of gap junctions maintain ionic continuity between adjacent cells and so allows information to travel between the cells.